Welcome to Educator.com. Today is the first in a series of lectures on cell structure and function. And in this lecture, we're going to discuss the two major cell types, prokaryotic and eukaryotic cells. We're going to begin by talking about cell theory. And cell theory states that cells are the fundamental unit of structure and function of living organisms. All organisms are composed of cells, and cells only arise from other cells. There are two major types of cells, prokaryotic cells and eukaryotic cells. Prokaryotic cells are bacteria. Eukaryotic cells include plant cells, animal cells, protists, fungi. Prokaryotic cells arose approximately 3.5 billion years ago, while eukaryotic cells arose more recently, 1.5 billion years ago. The endosymbiotic theory is a theory that accounts for the origin of eukaryotic cells from a symbiotic relationship between two early prokaryotic cells. Remember that symbiosis means a relationship between two organisms in which both organisms benefit. So the two organisms live closely together, they're independent, and they each interdependent, and they each derive a benefit from the other. Endo means within. So this is a type of symbiosis that involves one cell living within another cell. Early on, around 1.5 billion years ago, the environment greatly increased in the amount of oxygen. And that created a crisis for anaerobic organisms because oxygen was actually toxic to them. So what's believed to have happened is that prokaryotic anaerobic bacteria engulfed smaller aerobic bacteria. So larger anaerobic bacteria engulfed smaller aerobic bacteria. They each derived a benefit. The smaller bacteria received protection from the larger bacteria, and in turn, the smaller bacteria took care of the oxygen. So it actually used that oxygen to generate energy in the form of ATP. Eventually, over time, this relationship evolved for the two organisms to become one. And the larger bacteria, what was originally just a larger bacteria, would be analogous to the eukaryotic cell. And some of the organelles within the eukaryotic cell evolved from this smaller aerobic bacteria. For example, mitochondria and chloroplasts are organelles that have features that are similar to bacteria. And they provide proof for this theory. And we'll investigate those features more closely in a few minutes. In particular, chloroplasts are thought to have derived from the endocytosis of a photosynthetic bacteria called cyanobacteria or blue-green algae. Blue-green algae is sort of a misnomer because these actually are bacteria. They're photosynthetic bacteria, and again, it's believed that chloroplasts derived from these after the larger anaerobic bacteria engulfed them billions of years ago. Before we go on to talk in more detail about the cell types, we're going to discuss some of the tools and techniques used in the study of cells. One of these is, of course, microscopes. And the microscope was invented in the 17th century. Its use brought about great breakthroughs in biology because at last we could see much more detailed structures than we could with just the naked eye. Van Leeuwenhoek was an early inventor and developer of the microscope, and he was able to visualize microorganisms in a drop of water. He also visualized cells such as red blood cells and sperm cells. Robert Hooke described cells for the first time in a book in his book Micrographia that he wrote around 1665. When you consider microscopes, there's two main types, light microscopes and electron microscopes. Light microscopes are still widely used today, and they're the type of microscopes that Van Leeuwenhoek and Hooke used in their studies. Both types of microscopes, electron and light, are described according to two features. The first is magnification. Magnification tells you how much larger the specimen appears. So with a very high magnification, you can even go as high as 100,000 on an electron microscope. So the higher the magnification, the larger an image appears. And magnification could be as small as 2 times, 5 times, or 5,000 or even 100,000 times. With a light microscope, the maximum magnification only goes to about 1,000 times. Whereas an electron microscope, you can get a much higher magnification without the image becoming blurry. Okay, so magnification is the first characteristic that we use to describe a microscope. The second characteristic is resolution. Resolution doesn't have to do with the size of the image, it has to do with the clarity of the image. And resolution is defined as the minimum distance that 2 points can be together, 2 points can be apart, and still be distinguishable. So it's the minimum distance that 2 points can be apart and still be distinguished as separate objects. So it has to do with the clarity. If a microscope has a higher resolution, that means that you could see objects that are extremely close together, and they won't appear as one object. They'll actually appear separately as two objects. So the best microscope would have a very high magnification as well as a high resolution. Resolution is determined by the wavelength being used. Light microscopes use light passing through the specimen, whereas electron microscopes use a beam of electrons passing through a specimen. So an electron microscope is using something with a much shorter wavelength. And actually, shorter wavelength equals higher resolution. So that's a big benefit of electron microscopes. A disadvantage, though, is you can only use electron microscopes for dead specimens because the process to prepare the sample kills the cells. So they're only used for dead specimens. Whereas with an electron microscope, you can use either a living or dead specimen. Which is a big advantage. Now, electron microscopes come in two general types: transmission electron microscopes and scanning electron microscopes, or SEM. Transmission electron microscopes involve a preparation process for the sample in which thin sections are made and then visualized. So these are usually used to visualize the inside of the cell. We look at the inside of the cell. Whereas with scanning electron microscopes, we are generally looking at the surface of the cell, or the outside. With a scanning electron microscope, specimens are actually coated with a thin layer of gold and then observed. So coating them, or the fixative used for the specimens in transmission electron microscopy, all of these procedures are going to kill an organism or a cell, which is why you can only use these for dead specimens. Now, another difference between the two is that with TEM, the image looks very flat and two-dimensional. Whereas with scanning electron microscopy, the image looks, it has more depth, so more three-dimensional, 3D appearing, versus flat. And so that makes scanning electron microscopes very good to use with surfaces where you can see the depth of the different objects sitting on the surface of the cell. Another method that's used is cell fractionation, and that's what this picture is right here. And there are actually several steps to cell fractionation. The goal of cell fractionation is to separate out the different parts of the cell. With a microscope, you may be looking at the whole cell, you may be focusing in on a part, but here you can actually split up those parts and just study the mitochondria if you'd like. So there's 3 steps to cell fractionation. The first step is homogenization. Homogenization involves putting a specimen like this, which is actually going to be cells and they're in a liquid here, and you're going to put these in a blender. So you blend them up, but it's a type of blender and a type of procedure that is going to break apart the cells, which is going to release the organelles, but it's not going to actually damage the organelles. This creates a homogenate. So right here in this second tube is the homogenate. The homogenate is a suspension, so it's a liquid with all these organelles and particles and pieces of the cell suspended in it. The next step is to actually spin the homogenate. When you spin the homogenate, what's going to happen are the heaviest particles are going to sink to the bottom and form what's called a pellet. At the top are going to be lighter particles in a solution, and this is called the supernatant. So the supernatant is the fluid on top, the pellet is the solid part at the bottom. The next step is differential centrifugation. So differential centrifugation. So a very high-speed centrifuge called an ultracentrifuge is used, and you pour the supernatant into a new tube, spin at a higher speed than you used for the first time you spun. What that's going to do is allow particles that were too light to be forced to the bottom in that original spin to be forced to the bottom the second time. So you create another pellet and another set of supernatant. Pour the supernatant off into another tube, repeat the procedure. And you do this differential centrifugation until you have the pellet that you want. In the beginning, the pellets are going to contain heavier parts of the cell. By the end, you're going to have very light parts, such as the mitochondria or the ribosomes. Then scientists can take whichever pellet they are interested in, look at those particular organelles without having the rest of the cell right there. So again, the goal of cell fractionation is to separate out the cell into different sections, different organelles, and that's done by weight and by spinning them, and it differentiates each of these organelles and particles by weight. Finally, cell fractionation. Cell fractionation is a method of preparing a sample to use for transmission electron microscopy. In this, the cells are frozen in liquid nitrogen, and it creates a block. And then you fracture or split that block. And it splits right between the plasma membrane. So the plasma membrane has 2 layers. You could think of it as being like an Oreo cookie. And when you split it, it would split apart in the middle. So it splits right along the line of the plasma membrane. Next, that specimen that's been split is coated with gold or platinum, some type of metal, as well as a layer of carbon. And the specimen itself is digested away. So freeze fracture leaves you with the cast of the specimen, and you can then look at that specimen via transmission electron microscopy. Now that we've talked about methods used to study cells, we're going to go on to compare the 2 major cell types, eukaryotic and prokaryotic. So here we have a prokaryotic cell. This is a typical bacterial cell. For the eukaryotic cell, I've used a sketch of an animal cell. So this is a eukaryotic cell, but it is going to be a little bit different than, say, a plant cell, which we're going to look at in a moment. But we're just going to be talking about the general differences between prokaryotic cells and eukaryotic cells. So eukaryotes are found in the domain eukarya. whereas prokaryotic cells include the other domains. And remember that in the three-domain system, there's domain bacteria, domain archaea, so we'll say domains bacteria and archaea. And then domain eukarya includes plants, animals, fungi, protists. So prokaryotic cells, these two domains. Eukaryotic cells just include one domain, but it's a very large domain, the domain eukarya. Starting out by looking at the prokaryotic cell, you can see that it's much simpler in construction than the eukaryotic cell. Both prokaryotes and eukaryotes have a plasma membrane. This is also called the cell membrane, and it's composed of a phospholipid bilayer. So in the prokaryotic cell, the plasma membrane's right here. And in both of these, the plasma membrane is composed of phospholipid bilayer. In addition, bacterial cells have a cell wall. Animal cells do not have cell walls. However, fungi and plants do have cell walls. So some eukaryotes have cell walls. Animal cells do not have cell walls. The next thing to look at is where the genetic material of the cell resides. In prokaryotes, there is no true nucleus. Instead, what you have is called a nucleoid region. So this is the nucleoid region. The DNA is found here, and the DNA is circular. It is not organized into chromosomes, and it is not bound by proteins the way eukaryotic DNA is. So here, there is a true nucleus. This nucleus is bound by a double membrane, and inside the nucleus are chromosomes. These chromosomes are bound by proteins called histones. So chromosomes, and they're bound by proteins called histones, chromosomes with histones. The other thing you'll notice is that there are a lot more structures inside the eukaryotic cell, and these structures are called organelles, and we're going to talk in detail in the next section on subcellular structure about each of these organelles. But for now, just to name a few, these are the mitochondria, the endoplasmic reticulum, the Golgi apparatus, ribosomes. So eukaryotic cells have membrane-bound organelles that carry out specialized functions. Prokaryotic cells only have ribosomes. So both prokaryotic and eukaryotic cells have ribosomes. Prokaryotic cells are lacking the other organelles. So ribosomes here, and that's it for organelles in a prokaryotic cell. Another difference between prokaryotes and eukaryotes is that prokaryotes are mainly unicellular, while eukaryotes, like plants and animals, are very complex multicellular organisms. Prokaryotes also are smaller. They're about 10 to 100 times smaller than the typical eukaryotic cell, about 1 to 10 micrometers, whereas a eukaryotic cell will be in the range of, say, 10 to 500 micrometers. In addition to the nucleoid region, where most of the genetic material is, bacteria have structures called plasmids. And these are rings of DNA that contain just a few extra genes. Most of these aren't essential for survival. They often just help the bacterial cell to be more virulent. But this is another feature that prokaryotic cells have that eukaryotic cells do not. All right, so we've talked about a bunch of differences and similarities between prokaryotic and eukaryotic cells. Now to summarize those. Nucleus. Prokaryotic cells do not have a true nucleus. Instead, they have circular DNA in the nucleoid region. You'll sometimes hear this referred to as naked DNA, and that means that there's no proteins bound to the DNA. In contrast, eukaryotic cells have DNA organized into chromosomes. They're bound by histone protein, and they're contained within a membrane-bound nucleus. Organelles. Prokaryotes lack membrane-bound organelles, except for ribosomes. They do have ribosomes. whereas eukaryotic cells have many membrane-bound organelles that carry out specialized functions within the cell. Cytoskeleton. Since eukaryotic cells have a much more complex structure, in order to organize that structure, they have a system of microtubules and microfilaments that organize the cell via what's called a cytoskeleton. And we'll talk about that under subcellular structure, but for right now, just know that the cytoskeleton helps the cell to maintain its shape. It provides some protection, it provides organization, provides motility, whereas prokaryotic cells lack a cytoskeleton. Cell wall. Bacteria have cell walls, and these contain peptidoglycan. There are no cell walls in animals. Plants have cell walls composed of cellulose. Fungi have cell walls that contain chitin, and some protists have cell walls that are made of materials such as polysaccharides. Ribosomes. Both prokaryotes and eukaryotes have ribosomes, but they're different in structure. Prokaryotes have smaller ribosomes, and you'll see this S here, and it has to do with the sedimentation. Remember we talked about infractionation, that certain particles or organelles sediment to the bottom, they form a pellet, and that depends on how heavy the particular item is. So smaller ribosomes are found in prokaryotes, and those are 70S as far as their sedimentation, whereas eukaryotic ribosomes are different in structure. They're larger. They're actually 80S. Size. Prokaryotic cells are much smaller than the typical eukaryotic cell, which is larger. And then looking at it as a whole, not just as the individual cell, prokaryotic cells are usually unicellular, whereas eukaryotic organisms are multicellular. So prokaryotes are just one-celled organisms, whereas eukaryotes, plants, animals, very complex multicellular organisms that have different tissue types to carry out special functions. So the cells themselves become specialized. So we compared prokaryotic and eukaryotic cells. Now we're going to compare plant and animal cells. Remember that these are both eukaryotic cells, so they're going to have some features, many features that are actually the same. But there's very important differences too. And one of the central tenets of biology is that form follows function. This means that the plant cell is going to have some structures, so that's form, that'll carry out specialized functions. For example, photosynthesis. Animal cells don't carry out photosynthesis. They don't need structures that are going to achieve photosynthesis. So looking at these two in general, they both have a plasma membrane or cell membrane. Taking a look at plant cells, they also have a cell wall. Bacterial cells have cell walls, plant cells have cell walls, but in bacterial cells, the cell wall contains peptidoglycan, whereas in plant cells, cell walls contain cellulose. In fact, plant cells can actually have two cell walls. So they all have what's called a primary cell wall, and that contains cellulose, which is a polysaccharide. However, between the plasma membrane and this primary cell wall can be a secondary cell wall. So right in here, there might be another cell wall. And this secondary cell wall contains cellulose, but it also contains lignin. And lignin is a polymer that gives additional strength to the plant. For example, wood contains a lot of lignin. And because they don't have skeletons, plants need these cell walls to give them more strength, help them maintain their shape. In addition, the cell wall prevents the plant cell from bursting in a hypotonic environment. As we'll discuss when we talk about plasma membranes, when a cell is in an environment where there are less solutes in the solution than there are within the cell, then water is going to enter the cell. and that can actually cause a cell to burst. However, if it has a cell wall, it protects it from bursting. So the cell wall, in fact, helps the plant cell to maintain turgor or rigidity, because what happens is this structure, which is called a central vacuole, is something that is not found in animal cells. And water can be stored here, as well as food, and it takes care of waste. But one of the functions of the central vacuole contains water. And as the central vacuole becomes large, it can push on the cell wall, and that will make the plant rigid. So instead of wilting, the plant will stand up, the stem of the plant will be rigid, its leaves, and that's through something called turgor. So the central vacuole and the cell wall allow for turgor, which is pressure due to water pushing against this firm cell wall, and it allows the plant to maintain its shape and be rigid instead of just limp. Okay, so no central vacuole in animal cells, no cell wall in plant cells, in animal cells. So cell wall, central vacuole in the plant cell. Here in the animal cell, there's no cell wall and there's no central vacuole. So this is the plant cell. As you can see, a lot of the structures are similar because since they're eukaryotes, they both have a nucleus. It's membrane bound. You can see the mitochondria, endoplasmic reticulum, and various other structures. In addition, animal cells have structures called centrioles. Centrioles are regions that help to organize the cytoskeleton. They are found within centrosomes, and they play a key role in organizing what's called the spindle apparatus that helps to separate out the chromosomes during meiosis and mitosis so that the daughter cells get equal numbers of chromosomes. So centrioles are a type of microtubule organizing centers that are lacking in plant cells. So plant cells do not have centrioles, they do not have centrosomes, but they do have organized microtubules. So they have just a different type of microtubule organizing center. So plant cells, plants have no centrioles. They also have no lysosomes. Lysosomes are small sacs that contain enzymes that break down old organelles. Also, if there's an invader that a cell from the immune system has engulfed, such as a bacteria, lysosomes can break down those invaders. So lysosomes play a role in ridding the cell of things that are harmful or things that it doesn't need any longer, such as old organelles. Although the component parts of the old organelles are actually reused to make new organelles. Lysosomes are not found in plant cells. So no centrioles, no lysosomes. Plants are lacking those, but plants have a few things that animal cells do not. We mentioned the cell wall. I also mentioned the central vacuole. And another important part is chloroplasts. Chloroplasts are organelles that are specialized to carry out photosynthesis. And again, we're going to go into a detail of all these structures in the next section, but just to give you an overview that the major function of chloroplasts is to carry out photosynthesis. In addition, they contain green pigment. And that is why the stems and the leaves of plants appear green, because of this green pigment inside chloroplasts. To sum up, it's important that you know the differences between plant and animal cells because comparisons are important on the test. Okay, cell walls. Animal cells do not have cell walls. Plant cells have primary cell walls containing cellulose. Some plant cells actually have a secondary cell wall which contains both cellulose and lignin. Lysosomes. Lysosomes are present in animal cells. There are no lysosomes in plant cells. And again, these are sacs that contain digestive enzymes that break down old organelles and also pathogens. Chloroplasts. Animal cells do not have chloroplasts. Plant cells do. These are the site of photosynthesis. Centrioles. These are present in animal cells, not in plant cells, but plant cells do have MTOCs, which are microtubule organizing centers. So centrioles help to organize microtubules, whereas that role is taken on by different structures in the plant cell, although those have not been well described. Central vacuole. This is not present in animal cells. Animal cells can have vacuoles, but they don't have that large central vacuole. In the plants, these play a role in storage, waste disposal, maintenance of pH in the cell, so they're very important structures. In the beginning of this lesson, I mentioned the endosymbiotic theory, and I said that certain characteristics of eukaryotic cells, particularly the mitochondria and chloroplasts, provide support for this theory. So let's look at exactly what those characteristics are. So first, they have circular DNA. Bacterial cells have circular DNA, whereas eukaryotic cells have DNA organized into chromosomes. So the fact that mitochondria and chloroplasts have circular DNA is a similarity to prokaryotic cells, and that provides evidence for the fact that these may have actually descended from prokaryotic cells that were engulfed by other larger anaerobic prokaryotic cells. This DNA is, again, not in chromosome form, and they lack histones, which are the protein that DNA found in chromosomes is bound to. So this DNA looks a lot more like bacterial DNA in structure, in organization, than it looks like, rather than looking like eukaryotic organization of DNA. Another feature of mitochondria and chloroplasts is that they have a double membrane. And if you think about the endosymbiotic theory, it makes sense that organelles derived from the internal cell, the cell that was engulfed, would have a double membrane. So if you have a larger bacterial cell, and it has a cell membrane, and then you have a smaller aerobic bacterial cell, and this cell, the smaller cell, ends up engulfed by the larger cell. What's going to happen is you're going to end up with the larger cell with its cell membrane around it, and then the smaller cell inside with its original membrane. But as it pushed its way in, or was engulfed, the membrane from the larger cell wrapped around that smaller cell. So this actually, engulfed it, and it pinched off and formed an outer membrane around the original membrane of that smaller bacterial cell. So the double membrane found in both mitochondria and chloroplasts likely originated from the original membrane of the smaller cell and the membrane of the larger cell that engulfed that smaller cell. The third aspect of mitochondria and chloroplasts that provide evidence for the endosymbiotic theory are the fact that both these two organelles have ribosomes that are their own ribosomes, separate from those of the rest of the cell, and they are the same size as bacterial ribosomes. So recall that eukaryotic ribosomes are 80S. Here, prokaryotic ribosomes are 70S, and mitochondrial and chloroplast ribosomes are 70S as well. So another similarity to bacterial cells. Bacteria divide through a method called binary fission, where they split into two daughter cells. Chloroplasts and mitochondria also undergo binary fission. Mitochondria arise only from other mitochondria through binary fission, and chloroplasts arise only from other chloroplasts. Size. Mitochondria and chloroplasts are the size of a typical bacteria. So these 5 elements, the fact that both organelles have circular DNA like bacteria, they have 70S ribosomes like bacteria, they're the size of bacteria, they undergo binary fission, and they have a double membrane that probably originated from when they were engulfed. These 5 points all provide support for the endosymbiotic theory of eukaryotic cell origin. Now we're going to go through a few examples. So first, list 4 differences between prokaryotic and eukaryotic cells. First difference, prokaryotes. No nucleus, and the DNA is circular. Eukaryotes, they have a nucleus. The DNA is found within the nucleus of the cell. And the DNA is organized as chromosomes. The DNA is also bound to histones. So difference in the location and the organization of the DNA. Second difference, prokaryotes lack membrane-bound organelles. Except they do have ribosomes. So that is an important exception, except ribosomes. Eukaryotic cells have numerous organelles bound by membranes, which carry out specialized functions within the cell. Prokaryotic cells lack a cytoskeleton, whereas eukaryotic cells have a cytoskeleton that helps to organize the organelles within the cell, provides the shape and structure for the cell, and also allows for motility. Prokaryotic cells have smaller ribosomes. They have 70S ribosomes, whereas eukaryotic cells have larger ribosomes. They're 80S ribosomes. So I listed 4 here as the question asked, but there's certainly others that you could point out. Prokaryotic cells are smaller. So you could have discussed smaller versus larger eukaryotic cells. You could have mentioned the fact that prokaryotic cells have plasmids. Those are extra pieces of DNA that contain a few genes, and they are outside the nucleoid region. So this asked for 4, but there are certainly other differences that you could have listed. Example 2, describe the endosymbiotic theory of the origin of eukaryotic cells. Give 2 pieces of evidence in support of the theory. Okay, endosymbiotic theory. The endosymbiotic theory states that eukaryotic cells arose from engulfment of smaller aerobic prokaryotic cells by larger anaerobic prokaryotic cells. So remember, symbiosis means the 2 organisms live closely with one another and they each benefit from the relationship. Endosymbiosis means that one organism actually lives within the other organism. So one cell engulfed the other. They became more and more interdependent through time, through evolution. What was once an independent prokaryotic cell living within the larger cell became organelles. Two pieces of evidence. Well, the evidence has to do with the similarity of mitochondria and chloroplasts, which are organelles, similarity between those, and bacteria or prokaryotic cells. One piece of evidence is mitochondria and chloroplasts have their own DNA and it's circular. This is similar to that of bacterial cells. Mitochondria and chloroplasts also have double membranes. These double membranes may have originated from when the larger bacterial cell engulfed the smaller bacterial cell, and the membrane of the larger cell became the outer membrane of that smaller cell, which also retains its own membrane, which is then the inner membrane of the mitochondria and chloroplasts. This only asks for two. There are certainly other pieces of evidence. Size. Mitochondria and chloroplasts are similar in size to bacterial cells. The ribosomes of mitochondria and chloroplasts are 70S, like that of bacterial cells. They divide through binary fission. Both bacterial cells and mitochondria and chloroplasts divide through binary fission. So these are all pieces of evidence in support of the endosymbiotic theory. Example 3, state 3 differences between plant and animal cells. So I'm going to go ahead and write animal cells up here and plant cells up here, and then put what occurs for each. Cell walls. Animal cells have no cell walls. Plant cells have cell walls, which contain cellulose. They may also have a secondary cell wall, and I'll put this in parentheses because it's sometimes. So the secondary cell wall containing lignin. This is the primary cell wall. Secondary cell wall contains both cellulose and lignin. Animal cells have lysosomes. Plant cells do not. And remember that lysosomes contain enzymes that break down old organelles or pathogens or whatever a cell needs to destroy. In addition, animal cells have centrioles. These organize the microtubules, particularly during meiosis and mitosis. Plant cells do not, although they do have microtubule organizing centers, MTOCs. So that's 3 differences. Another big difference is that plant cells have chloroplasts, which are the site of photosynthesis. Animal cells do not. Plants also have a large central vacuole, which carries out multiple functions such as storage of water and removal of waste products. Animal cells do not. So 3 differences for the question, and there's a couple more major ones as well. Example 4, describe the process of cell fractionation. Okay, cell fractionation has 3 steps. The first is homogenization. In this step, the cells are broken apart, but it's done in such a way that the organelles are not damaged. The second step is to spin the homogenate. So the homogenate is a liquid that contains the various parts of the cells in it. Spinning the homogenate is going to result in a pellet at the bottom of the tube and a supernatant suspended on top. The supernatant is poured off, spin the homogenate, and then pour off supernatant into another tube. Spin that tube. pour off that supernatant, spin that tube, and keep going at faster and faster speeds to separate out lighter and lighter organelles. So this is called, this process of pouring off the supernatant, spinning it, pouring it off, raising the speed, is called differential centrifugation. And it allows you to separate out these parts of the cell by which pellet they sediment into. Why is it useful in the study of cells? Well, cell fractionation separates organelles into different pellets. The early pellets, those are ones that were spun, the tube was spun at a lower speed. Those are going to be heavier items. The later pellets, the ones that were spun very fast in order to get them to sediment, are very light. They're things like mitochondria and ribosomes. So that concludes this section of educator.com, our discussion of the differences between the major cell types. See you again soon.